Receptor proteins are like smart doorbells or sensors on the surface of cells. They detect special chemical messages, called ligands, coming from outside the cell, like hormones, neurotransmitters, growth factors, odors, light, or drugs, and then tell the cell, "Hey, something important arrived." This starts a chain of events inside the cell, signal transduction, that leads to some response, such as muscle contraction, gene turning on-off, smell perception, vision, etc. Now let's understand step by step in very simple language. There are about 20 different families of receptor proteins, and each family has several slightly different versions, isoforms, that can respond to different ligands. Imagine 20 big receptor companies. Each company makes several slightly different models of doorbells. Each model is tuned to recognize a specific type of visitor, ligand. Most of these receptors are transmembrane proteins, meaning they sit in the cell membrane and have parts both outside and inside the cell. Receptors in the same family share similar structural features, either the outside part, extracellular domain, binds similar types of ligands, or the inside part, cytoplasmic domain, uses similar ways to send the signal inside, or both parts are similar. Family members look alike in important regions, either the guest recognition area, or the internal messaging system, or both. Important concept. No strict rule. There is no clear connection between what type of ligand it is, small molecule, protein, light, odor, etc., and which receptor family it binds to. Also, the same ligand can trigger very different receptor families in different cells, different signaling strategies inside. Example: adrenaline, epinephrine, can bind to different receptor types in heart cells versus liver cells, different effects, heart beats faster versus sugar release from liver. This means nature mixes and matches freely. Same chemical signal can produce completely different results, depending on which cell and which receptor it meets. Main grouping of cell surface receptors. They are usually divided into 3 big categories, based on how they're connected inside the cell. There are also some receptors that don't neatly fit into these 3 groups. Special focus on G-protein coupled receptors, GPCRs. All known GPCRs belong to one big structural family. They cross the membrane 7 times, like a snake winding in and out. That's why called serpentine, 7-pass, 7 transmembrane, or 7 helix receptors. The transmembrane parts are mostly alpha helices, coiled like a spring. This family is very ancient, found in almost all eukaryotes. Famous examples. Odorant receptors allow you to smell thousands of odors. Rhodopsin, the light-detecting protein in your eyes, turns light into electrical signal. Many receptors for medicines or drugs, for example, beta blockers, antihistamines, painkillers, antipsychotics, etc. Around 30 to 50% of all modern drugs target GPCRs. Quick summary for students. Receptors are like cell surface sensors for external signals. About 20 families, most are transmembrane. Same family, similar structure in binding or signaling part. No strict rule, one ligand, many receptor types possible. Same receptor family can bind very different ligands. Main 3 types. Ion channel, fast, direct ion flow. GPCR, 7 times through membrane, uses G-protein, huge family, smells, vision or drugs. Enzyme-linked, receptor has its own enzyme activity. GPCRs are the celebrities. 7 membrane crossings, very ancient, very important clinically. This flexibility is why our body can do so many different things with a limited set of signaling molecules. So, this is it for today. I hope you liked the video. Please subscribe this channel and press the bell icon.